younger and younger people with CF, and a drug development pipeline with more programs in it than any previous time in our history. Of course, a big part of this is that we want to be able to treat the underlying cause of the disease for all people with CF, no matter what mutations they have, which is why we continue to boldly advance our Path to a Cure initiative, the $500 million commitment to draw the world's best science, tools, and technologies to find a cure for CF. We're funding collaborations with leading biotech and venture capital firms, which have already resulted in 35 major industry research programs focused on Path to a Cure genetic science since we announced the initiative. These include RNA therapy, gene editing, and other technologies you'll hear about today. This work is not just for someday, though. Several clinical trials for CF genetic therapies are already underway or will begin in the next 18 months. To meet the changing and diverse needs of all people with CF, we also continue with our work to address the challenges of CF today, including infection, GI issues, and lung transplantation. And our care model continues to evolve, including increased focus on improving health outcomes and better addressing health inequities. There's so much work to talk about, but I'm not going to dive into the details now. During today's plenary and over the next few days, you'll be hearing the details of all this work, and we'll understand why there is so much to be excited about as we all work together to continue to write the amazing CF story. Before we head into the first plenary, I need to take a moment, though, to recognize someone who has played a prominent role in writing this amazing story, a leader whom so many of you know, and that is Cam McLoud. This spring, Cam stepped down as chair of the Foundation's Board of Trustees after more than 2 decades in this role and more than 35 years of service on the board. And we welcomed our new board chair, Casey White. Casey is the first person with cystic fibrosis to lead the Foundation's Board of Trustees, and she is well prepared to help us build on Cam's powerful legacy as we enter a new era in CF. We're grateful that Cam will continue to serve as a member of the board into next year and allow us to benefit from her wisdom and experience. You know, as chair of the board, Cam regularly took part in research conferences, attending as many sessions as possible, taking notes, jotting down questions. We've all been on the receiving end of those questions before. Cam has a special and lasting devotion to advancing, or to all of us who advance CF science and care, to all of you here in this room. And we know that her passion for learning about scientific advances was based on her determination to do all she could to help her son Will and every person living with CF. So as we've pursued bold and innovative strategies to accelerate the search for a cure, Cam has provided steady support and generous counsel to me personally, to the board, and to the Foundation leadership team. And so Cam, because of you, we're in a great position now to go further and faster than ever before. We thank you. Now, I'd like to share a small token of appreciation for what from the Foundation team for all you've done, Cam. So I'm just going to invite you, Cam, to come on out.
Am I going to speak after that? Thank you. Thank you, Mike, and thank you to the team for this precious gift. How special it is to be here with all of you today, and that includes many of you joining virtually, especially those of you with CF. You are at the heart of everything that we do. It's been such an honor to serve as chair of the Foundation's Board of Trustees and to be part of this community throughout a time of amazing progress in CF. All of you here are extraordinary care teams, dedicated scientists, fellow trustees, and passionate volunteers, have helped make this progress possible. I first engaged with CF community almost 40 years ago as the mother of a child diagnosed with CF. I quickly linked arms with other parents and families, and this wonderful community became my family, my CF family. Those early days were not easy. We didn't have the science to target the basic defect. In fact, the gene had not even been discovered. But that did not deter us. Some of my best memories of serving on the board and later as chair go back to when the foundation resolved to make the bold investments needed to accelerate our pursuit of a cure. Those first steps led to the scientific breakthroughs and the many exciting moments that followed. And today, we're able to pursue genetic therapies for all people living with CF. Something unimaginable not too long ago. This is especially important to me because my son Will had 2 nonsense mutations and was among the 7% who don't have a modulator treatment yet. I thank all of you from the bottom of my heart for all that you do every day to make the difference in the lives of people with CF and your tireless commitment to working together until it is done. We know we still have important work ahead, and that's one of the reasons that we've gathered here in Philadelphia, to look toward the future and focus on how we can do more. Today, it's my great honor to introduce one of the people who will be leading our important efforts as we take on the challenges in the years ahead, my colleague and my dear friend, Casey White. I could not imagine a better person to lead the Foundation's Board of Trustees at this exciting time. Casey embodies the qualities that we will need to get us to our ultimate goal: vision, fortitude, and exceptional commitment and drive. I call this a dream come true, that a recognized leader, someone with CF, is chairing the board and propelling us down the path to the cure for all people with CF. I look forward to seeing all that will come in the future and to being part of this amazing family until it's done. Now, please join me in welcoming Casey to the stage.
Thank you, Cam. And I would like to share my personal gratitude for your friendship and leadership and longtime dedication to helping every person with CF fulfill their dreams. It is truly an honor to be with all of you this afternoon, and I'm very excited about attending many of your sessions throughout this conference. I also send my warmest welcome to all of you joining online today. I am in awe of the exciting research moving forward in CF and the steady improvements and innovations in care. You are transforming lives, and we thank you. When I was diagnosed with CF at the age of 3, this was a disease of little hope. The gene had yet to be discovered, and the life expectancy was in the teens. However, the doctor that diagnosed me helped my parents envision a different outcome. She told them to raise me to be a responsible adult because she had faith in the CF Foundation's mission, and she was encouraged by the research on the horizon. This simple but powerful message allowed my family to choose hope over fear. Over the years, there were ups and downs, but it seemed like every time my health started to decline, there was a newly approved treatment for CF that kept that decline at bay. First, Pulmozyme, later Toby and Cayston. And these treatments made it possible for me to get married and be a mom and a coach. But in my 30s, my health took up a larger part of my life. I had more treatments, more frequent hospitalizations, and I had to retire from coaching. And my future seemed uncertain, and I was desperate for more therapies because I wanted so badly to be able to be there for my husband and son. It was around this time when I attended NACFC for the first time, and I distinctly remember looking all around me to see a room filled with people who spend their days finding the answers that I and so many others needed. And I was filled with this sense of hope that has stuck with me ever since. I pictured this room when I was at my sickest to give me strength. I pictured this room with such gratitude when I celebrated my 40th birthday a few years ago. I pictured this room when my son turned 16 this past August, which was my life expectancy when I was diagnosed. And I especially pictured this room when I took my first dose of Trikafta, and also a month later when my PFT showed that my lung function had improved by 15%. Thank you. And I pictured this room when I was able to return to coaching and also complete a master's degree and still more, accept my new role as the chair of the Board of Trustees. I am living proof of the way you all are transforming lives. But as Cam mentioned, we all know that there is more to do. And when I think about our challenges ahead, I will picture all of you in this room today. You are the ones who help us navigate the complexities of living with cystic fibrosis. And I know you will continue to find the answers for those who don't benefit from the current modulators, for those who are waiting for novel antibiotics to treat drug-resistant bacteria, for those struggling with manifestations of CF outside of the lung, and for those who are post-transplant. And this is the room that will find genetic therapies that will bring a cure for every person with CF. You are our hope and action and the reason we can continue to dream. And for me, that dream includes being there for my son's milestones and maybe even holding my grandchildren someday. And this room extends to those joining us virtually as well. And I would love for the people online, especially those with CF and their families, to be able to see what I see, a room filled with dedicated researchers and clinicians and CF Foundation staff. So if you could please stand in just a second and face the cameras in the back of the room. They're towards the center, but the cameras will pan to get a broader view, so the people online can be filled with hope as they see your amazing faces. And as a coach, I know that the best way to get ready for a big game, or in this case, a big conference, is a team cheer. So I thought we could say together on my count, until it's done. So if you would stand up and face the back of the room, that would be amazing. The lights are coming on. And we're only going to cheer once, so we have to go all in, loud and proud. All right, so until it's done on 3. 1, 2, 3. Until it's done. Thank you all for being here today. I look forward to working with you until our job is done. Thank you.
Thank you, President. Well, thank you, Casey. That was an absolutely perfect way to kick off the meetings. And I can tell you, Casey, that everyone in this room and online is committed to this work for people with CF until it's done. So thank you. Now I'm excited to introduce the first plenary, which focuses on how we are addressing the needs of all people with CF, and specifically those with nonsense and rare mutations who currently lack a treatment for the underlying cause of their disease. This topic speaks to the heart of our mission. We want every person with CF to be able to live a long, healthy life, and we will not leave anyone behind. As you're going to learn, we're continuing to make progress not only in expanding modulators to those with rare mutations through theratyping, but also in our work to advance genetic-based therapies, which can benefit anyone and everyone with CF. Today, we'll hear about this progress from 3 exceptional medical leaders. Let's start with someone most of you know, and that is our new Chief Scientific Officer, Dr. Stephen Rowe. Steve came to us as a professor and CF Research Center Director at the University of Alabama at Birmingham and brings more than 2 decades of experience working in CF. He is a pioneer in personalized medicine and translational research for CF and served as a principal investigator for the first triple combination therapy, Trikafta, which has been transformational for so many people in our community. Steve has been the lead scientific advisor to the Foundation for a decade and stepped fully into his position as CSO in July. Steve, we're thrilled that you're on board and that you're leading the charge as we build on our legacy of scientific innovation. This afternoon, Steve's going to be focusing on nonsense mutations and innovative research underway to help enable the production of functioning CFTR protein. Next, we're pleased to welcome Dr. Michelle Hastings, who will discuss research into the development of a novel approach using antisense oligonucleotides, ASOs, to treat individuals with splice mutations. Michelle is a professor and director of the Center for Genetic Diseases at the Chicago Medical School at Rosalind Franklin University of Medicine and Science. Her research focuses on understanding the genetic basis of disease and discovering new therapeutics that modulate the process of pre-mRNA splicing to alter gene expression. Her work has resulted in the discovery of effective means of targeting splicing with antisense molecules for the potential treatment of a number of neurodegenerative diseases, including Batten's disease, Usher syndrome, Alzheimer's, and Parkinson's. Dr. Hastings has many patents for her discoveries, and in 2019 was recognized as a researcher to know by the Illinois Science and Technology Consortium. And then to bat third on the lineup to bring us home will be Dr. Rebecca Darrah. She's going to delve into the latest research in mRNA therapy, which may prove to be an important new treatment for people with CF that aren't able to benefit from CFTR modulators currently. Rebecca's a board-certified and licensed genetic counselor, an associate professor in the Department of Genetics and Genome Sciences at Case Western Reserve University, where she's also the co-director of the Genetic Counseling Training Program. There, she leads a research lab that focuses on genetic modifiers of CF airway disease, including study that relationship between variations in specific genes and clinical severity in CF lung disease. Rebecca's lab also uses mouse models of CF to study interventions aimed at improving pulmonary mechanics, response to infection, and circadian rhythms as a means to improve quality of life for people with CF. So with that, I'd like to welcome Dr. Steve Rowe to the stage. Steve, we're excited to hear your remarks today.
Well, thank you, Mike, for that introduction. I'm so excited to be back here with you. It's energizing to return in person, and I know you too feel the enthusiasm brought forth by the conference. I've been in the field as a clinician and as a scientist since I first committed to help people with CF as a counselor for children with special health needs. And so to the many of you who I have not yet met, I wanted to share where I stand, what brought me to this new role after 20 years as a clinician and as a researcher. When I started treating adults with CF at a children's hospital, the entire focus of, in one of the words of one of my favorite people with CF, was slowing the racing train. And I also remember when we first tested as the molecule known as VX-770, the remarkable effects of the CFTR modulator therapy and when they were first realized. How, working with our team, we could flip the switch from somebody who had CF to someone who seemingly had just a bit less than that. And how a person with CF could recognize the difference from a bad CF cough and a healthy cough, bringing them to health for the first time. And this showed us what could be done, and in fact, the last few years have brought that to fruition for many. But for me, despite our collective success, the job feels very much unfinished. And I know that every one of you that made the effort to be here remain totally committed to seeing the job to completion. And I'm inspired by our recent past and the foundations on which we stand. I believe our community is poised to bring these sorts of transformative treatments for all. Our ultimate goal, of course, is to help shepherd in a new era of medicine that we're beginning to see in other diseases. And when it is now possible for people with a genetic cause of blindness to have a single injection in their eye and at the end of the day be able to read the newspaper, I'm absolutely convinced that CF should help lead the way as we do the same for the far more challenging task of treating a multi-system disease that affects the lung. And as we look to the future, we stand on a strong foundation on which to accomplish our task. We're at a crucial moment in the field of CF. While the pace has never been faster, as you can see by the uptick in slope on the right side of the curve, we must seize on this momentum. As we'll hear from Plenary 3 speaker, Dr. Liz Tullis, the median age is improving fast and now accelerating at a pace that exceeds 1 year for every year lived. And we can also see the number of transplants dropping dramatically, and pregnancies, something I once as a clinician would recommend certain individuals to avoid, is now on the rise, and for mostly all the right reasons. And I know that you will be excited to join me for our Plenary 2 speakers, Dr. Perlanini and Hamblett, who will describe how the pipeline of therapies and the tools to advance them is as rich as ever, but isn't without challenges that we will have to navigate together. As we look upon the current landscape of highly effective CFTR modulator has made a foundational change in the current outlook of people with CF. First with ivacaftor or Kalydeco for gating mutations, and now elexacaftor, tezacaftor, ivacaftor for Trikafta. For those with F508del and beyond, we're now encompassing genetically defined up to 94% of individuals. Modulation of CFTR to address the basic defect has altered the outlook for so many, even if it has not affected everyone in the same way. And now we need to turn our focus to those at the top of the curve who have not yet been able to access these medicines, either due to the nature of the mutations or perhaps the continent where they live. Well, one thing we can be proud of about our community is that we have learned from both our successes and our disappointments. And it is our recent experience with these CFTR modulators that have provided a clear roadmap for how to tackle the challenge before us. On the x-axis, you can see CFTR function that can be attained in the laboratory, guiding data for us as we develop new medicines, along with, on the y-axis, clinical benefit as measured by the change in FEV1. And what you can see is that we're getting better over time, with improvements in the CFTR modulator combinations with greater and greater efficacy as we accomplish more and more individuals. And we've also learned that these benefits have been substantial, including both the expected benefits in respiratory health, but also changes in well-being, growth, and nutrition, and even the remarkable possibility of organ function preservation in the pancreas. Accompanied by data in the registry, we're also getting clear evidence of improved survival, an amazing accomplishment for this chronic disease. Clear evidence that we can, for a fact that we can almost take for granted. We now know the target and how to hit the bullseye. If we can restore CFTR function for all, we can expect a pronounced benefit for people with CF. Well, the broad utilization of these medicines and the notion that we continue to improve them provides a major impetus to continue to understand this new landscape. We need to define the current and emerging challenges for people with CF, determine what is addressed, and where we have more work to do. To do this, we've embarked on major research efforts to define the new normal, enrolling dozens of studies shown here on this slide, with a total number of participants that now exceeds over 7,000, with others that are not even shown. And I'm particularly excited to hear throughout the conference the results of important studies such as the Simplify study, our first data-driven attempt to help simplify the regimen for people with CF and determine what is needed for the future, and the Mayflower study to help understand our evolving landscape of pregnancy in CF. We'll also learn from the microbiology results of the PROMISE study and where we might have more work to do. And I want to mention the CHECK study as well, where we're involving over 5,000 individuals that seeks to establish sweat chloride as the blood pressure equivalent for people with CF and their providers, perhaps allowing the selection, optimization, and even expansion of CFTR modulators with drugs upon the horizon. But in the midst of this success, we do not want to lose sight of the reality. We have more work to do, and we have not achieved our goal of a cure. Here you can see plotting time since initiation of ivacaftor for people with the G551D and similar gating mutations, that lung function still declines at an unacceptable rate. We also know that people with non-CF bronchiectasis, despite having CFTR function that's near normal, they continue to have progression of their disease. All this is to say that modulators have helped, but they are not a cure. To address the continued needs of people with CF, we're going to need new partnerships across respiratory diseases that have commonalities with CF to help facilitate drug development and address all of the needs of our patients. In talking about the future, I would be remiss not to mention the path to a cure. Our largest and most ambitious initiative yet, with a goal to deliver the ultimate functional cure. And you should know, from of course introduced by Mike, that we are just getting started with our total allocation and doing so in a diverse array of ways. This slide shows the breadth of our initiatives and highlights we are doing so in a number of innovative ways, partnering with companies and platforms, bringing them early into the CF process so they remain committed until its completion. And what you can expect is continued emphasis in this area as we recruit the best and brightest to the field from academia and from industry and help combine them with those of you in enormous talent in the room who have a focus on CF and bring with our collective new advancements for people with CF. And although gene editing and gene delivery and other curative treatments is a major focus, we know this is a long-term project. We've had disappointments before, and we are likely to have them again, but know that we are in it for the long haul. I'll know you'll be excited to learn the emerging results of DNA studies already underway, our first of many ambitious results to follow. In fact, we've had our first glimpse of these data from when presented by Jennifer Taylor-Kusar earlier today. But today we will focus on these efforts of mRNA restoration, splice correction, and nonsense mutation read-through that we hope present near-term opportunities to improve the lives of people with CF and address these mutations. Of course, the focus has been upon the progress we've made toward restoring CFTR in the near term for mutations has so far been difficult to treat, and where current modulators have not been on the immediate horizon. Those at the top of this curve. And if you look at that carefully, clearly we have more important work to do as we focus upon these individuals. Including in that is for missense mutations and in-frame deletions, including more new theratyping efforts and multiple new modulators currently in late-stage developments with novel mechanisms of action can help cover more and more of these mutations, especially when coupled with theratyping efforts. And you'll hear from our other presenters, Dr. Hastings and Dr. Darrah, who will approach ASO and mRNA therapies for the splicing variants and frameshift mutations, while I will focus on nonsense mutations, which all of you in the room may know has been one of the toughest nuts to crack. I began my first research work in CF in this area, having recently been conceived by David Bedwell and then mentored by Eric Sorcher and J.P. Clancy. And with that work in cells, we subsequently expanded to some of the first studies in humans using the nasal membrane as a learning center. Studies that were later successfully completed by Drs. Wolshansky and Karem in Israel, showing the proof of concept that indeed aminoglycosides could restore function for people with nonsense mutations. But we also tested PTC-124, the medicine that became Adalurin, and struggled with the need for more precise outcome measures and suffered from the inconsistent results of a marginal therapy. But I hope you'll take heart that we've learned so much since then. Given where we are today, I believe that there's real optimism, real reasons for optimism, and that we are beginning to crack the code for what it takes to develop an effective medicine, and that we will show you that there's clear evidence now from the laboratory that we can now achieve this in important and predictive model systems. And while there's much to be done to make these a medicine, this is a key foundational step that puts us on a clear path in these areas. To consider the urgency and how this is felt on an individual level, let's hear from the McClain family that describe their predicament.
I'm Adrienne McClain. This is my husband, Jordan McClain. We live in Colleyville, Texas, and we have two little girls. Carly is 9 years old, and Abigail is 6 years old, and she has cystic fibrosis. Abigail was diagnosed when she was 5 weeks old. She started doing breathing treatments regularly before she started brushing her teeth regularly.
Abigail's biggest challenge is that she just doesn't get over colds or flus like most kids would.
I have a bunch of coughs and it doesn't feel good.
She and her sister both do a Pee Wee cheer team. We had a game that was in the middle of the afternoon, really hot. She was sweating quite a bit, and we ended up leaving the game early. We have to pay really close attention to Abigail and notice when she needs to take a break.
Living with the disease is a process. Abigail has 2 rare mutations which produce premature stop codons.
Abigail is not eligible to take modulator drugs because of the mutations that she has. I don't think it's something that we dwell on every day. You know, we have a first grader and a third grader who are busy with school and activities, and I think that we're just trying to let them live their lives and do her treatments regularly to stay on top of her health.
I'm very hopeful that there will be new treatments that come out for Abigail. I think our hopes and dreams are like any other parents' hopes and dreams, and that we want to see not just Abigail, but both of our girls thrive into adulthood and get to live out the lives that they choose to live.
When I have been at different events for the CF Foundation and met parents who have adult children with cystic fibrosis, I'm so encouraged because they have walked this journey for many years, and seeing their adult children thrive really made me realize that while my dream is to see Abigail graduate from high school and go to the college she wants to go to and maybe have children of her own one day, I realized that Abigail's dream is going to be seeing all of those things for her children. And so she wants to live a life where she gets to do all those things and then see her children do those things. So that's what I really want for Abigail.
If I were to say anything to people that are working on CF, researchers, scientists, it would be don't give up, because our daughter's life depends on it.
To all of the researchers and scientists, I just think we're so grateful. There's both excitement and just a hope that this powerful, strong community will stay in the fight for us. Just having that hope that the effort, the advocacy, the fundraising, all of the things that it takes to develop these life-changing drugs keep happening until we have a cure for Abigail and for everyone living with rare and nonsense mutations.
Well, thank you to the McClain family for sharing their story. I can certainly relate with my team treating the dozens of patients with nonsense mutations, including those that travel from elsewhere as they attempted some of these clinical trials. It's not hard to be motivated by the continued need, and we have much to do to accomplish our task. It's like the development of CFTR modulators. Our approach has always been based on a strong understanding of the science and the molecular mechanisms at play. So let's dive into that. Here you can see in this slide the molecular mechanisms of translational read-through that starts with a premature termination codon in the DNA, shown in red. Through transcription, this forms a mRNA that has a stop sign or a stop codon right where it shouldn't be, in the middle of the CFTR transcript. Through splicing, a mechanism you'll hear about in more detail from Dr. Hastings, this mRNA transcript is moved out of the nucleus and into the cell where translation begins. But instead of a full-length CFTR protein being produced, because of that abnormal stop sign, or the stop mutation, a truncated protein results. Well, this moment where the ribosome is translating the mRNA and reaches the stop codon represents a pivotal moment for the cell, a moment of decision in which we have the opportunity to tip the scales. By far the majority of the time, translation termination occurs, recruiting in release factors such as ERF1 and ERF3 that we'll talk about shortly, and terminating the protein. But a small fraction of the time, perhaps 1 in 1,000, instead a near cognate tRNA is brought in. This is the tRNA that matches the stop codon, but instead has an amino acid attached to it and allows the translation to continue. We need to alter the balance of the cell to be able to bring this therapy to the fore. And this is a really important concept. So let's review it again in this slide. Here again, when the ribosome is normally translating the mRNA, when a normal vacancy occurs, a tRNA is brought to the fore and a CFTR protein production continues. However, when the step is reached where the stop codon resides, instead there's an equilibrium, and release factors are brought in the majority of the time. This causes translation to terminate, and further, the mRNA to be degraded because of the cell senses this nonsense mutation. A non-functional CFTR protein results, and in few numbers. The scale is firmly tipped against therapeutic benefit. Well, how might translational read-through work? Well, here we can see if we could bring a small molecule, in this case represented by the go sign, it can interact with the ribosome or the mRNA or the key aspect of this translational machinery. Alter the balance so that instead near cognate tRNA is brought in more of the time. A protein production can continue, resulting in a full-length CFTR protein. And this can go on to transport chloride as we need it to, although in some cases not quite as well as a normal channel. We'll talk about that shortly. In this way, we can alter the balance, bringing translational elongation or translational read-through happening more of the time for therapeutic benefit. We want to tip the balance in our favor. And with this in mind, there's now 2 decades of research showing that various agents, initially with aminoglycosides, but now many other experimental drugs found by many different parties that are effective in this fashion. However, we've also learned, even as recently as the last 6 months, when the aminoglycoside ELOX02 was not successful in restoring sufficient CFTR function to confer benefit, that we might need to think beyond one single molecule on one single mechanism to achieve our goal. But this, of course, is not uncharted territory for us in CF. We have been here before. In this rendering, we can imagine therapies as building blocks, stacking together to achieve the clinical threshold to confer substantial clinical benefit. For the G551D mutation, this was more straightforward, as a single medicine was able to achieve this task. And as you know, for the F508del and other related mutations, this was much more challenging. I remember at this venue where we had a great deal of hand-wringing when 2-drug therapy did not achieve the results we desired, and ultimately had to renew screening efforts to ultimately find the 3-drug combination that we now know as Trikafta. Now we have the tools to do the same for nonsense mutations, but we have not yet brought them to fruition. We'll examine the data that shows that this is becoming a reality in the laboratory and achieving the results necessary to imagine a therapy with this approach. Let's look together how we might break the molecular biology down into steps that can lead to this multi-agent therapy. In health, DNA leads to mRNA, the full-length protein, and anion transport. However, in nonsense mutations or in disease, that abnormal stop codon results in the absence of the full-length functional protein, as you can see in the middle of the slide. Further, the nonsense-mediated decay process reduces the number of mRNAs that are available. And finally, the anion transport is also dysfunctional because most of the time, or some of the time, the amino acid that's inserted is not exactly the normal match for the sequence. So how can we address this with therapy? For the one, we can bring translational read-through, as I've already described, to generate the go sign to generate full-length CFTR proteins. We can use approaches to augment the level of mRNA to inhibit nonsense-mediated decay to increase the number of mRNA transcripts, ultimately resulting in increased number of CFTR functional proteins. And finally, we can use what we've become familiar with, and that is CFTR modulators to augment the function of those CFTR channels. What I'll show now is that what I hope you can understand with this is we're very much like when we broke the glass ceiling for the F508del CFTR protein, when the combination of 2 correctors and potentiator could indeed rescue the high levels of CFTR function. And it's my belief that if we take the same approach, we can stack the scales in our favor, and we will be successful. Well, the first such approach that's in the laboratory that I want to introduce you to is how to make this existing read-through therapy better. Again, capitalizing beyond single drug therapy, we now have mechanisms that can deplete these polypeptide release factors. In this case, ERF1, tipping further the balance to allow translation elongation and read-through. Well, this has actually been achieved in an important publication in the last year by my colleagues at Southern Research and investigators at UAB, where we showed that a new molecule, SRI-41315, a tool molecule, was able to deplete ERF1 levels. You can see that as it designated in the red box, where the column with SRI-41315 reduced the amount of ERF1 present as compared to the G418 or DMSO control. And further, when we brought this molecule, or its relative molecule, SRI-41765, which even has more favorable pharmacologic properties that can be, that we envision can be ultimately made into a drug, and tested this with non-organoids, the organoid cells that you had introduced before at this conference that swell upon the rescue of CFTR, that when we added SRI 41765 to ELOX-02, the aminoglycoside, you can see those gold bars were very much higher than the monotherapy lanes, and reaching to levels of CFTR function that we expect might make a clinical difference for patients. Furthermore, in a concept that again I've introduced, if we add correctors and potentiators to the mix, in this case elexacaftor, tezacaftor, ivacaftor, the green bars are even higher, again in a range that we expect to be able to make a difference. The story does not end here. You can read about this in poster 645, and also in a very important publication by Lee and colleagues, where Scott Randell's group showed the same sort of concept can be achieved with ERF3 inhibition and CFTR function restored into the clinical range of the laboratory, including those for the W1282X mutation, with just this approach alone. Well, the story does not end with just small molecules. We also have the approach of ACE tRNAs, where it can restore CFTR expression and function to a variety of nonsense mutations. These are anticodon engineered, or ACE tRNAs, that are specifically designed with RNA transcripts to help outcompete these release factors. When we can, instead of release factors being recruited to the stop codon, we can introduce an ACE tRNA that can outcompete and allow a protein production to continue. Well, this was conceived by Chris Ahrens and John Look, who have shown this to be effective. And in fact, if we look at the laboratory evidence, you can see that across 3 different important premature termination codons, the most common that affect people with CF, G542X, R1162X, and W1282X, shown in orange, red, and blue correspondingly, that those cells treated with these ACE tRNAs achieved substantial levels of CFTR mRNA levels, and consequently not shown here, protein levels and function that can begin to approach that seen in the untreated non-CF controls on the left side of the screen. The analogy of this approach is that we can build a Trojan horse for good, in which the tRNA, specifically engineered for read-through, can outcompete these release factors and restore CFTR function. Well, a second important approach is the inhibition of nonsense-mediated decay that can restore mRNA levels of nonsense mutation. Nonsense-mediated decay is a process in the cell that can deplete the mRNA levels when it's present with a premature stop codon. And what happens in the cell is that there are particular proteins that target these mRNAs for early degradation. The most important one for CF is SMUG6. So if we can deplete SMUG6, instead of the mRNA being depleted, we can expect an acceleration in the number of mRNAs that are available for translational read-through. And in fact, in data generated by Adrian Craner and also the CFFT laboratory, it's been shown that this SMUG6 ASO, a molecule that's a nucleotide that you'll hear more about from Dr. Hastings, can then be combined with the aminoglycoside G418. And in the yellow bar, you can see that more than doubles the effect seen with the aminoglycoside alone, showing that by inhibiting the mRNA nonsense-mediated decay to augment mRNA levels, then we can have a meaningful addition to CFTR read-through therapy. And the third approach is one that you're familiar with, and that is the use of CFTR modulators that can be targeted for read-through to augment the read-through product further. When we have translational read-through, there's the truncated CFTR may become full length, but sometimes the wrong amino acid is inserted. While the CFTR is full length, it may not have the same function because a missense mutation results. Well, we can address this by individualizing the selection and testing of CFTR modulators to get the most function out of these novel mutant channels. This has been shown in the laboratory to actually be effective, and as we march down using systematic approaches to identify the amino acids that are inserted. In this case, we used mass spectroscopy to identify the different amino acids that are inserted at the important 1282 position upon read-through with an aminoglycoside. And instead of the tryptophan, or W, shown in red, which is the native amino acid, most of the time, other amino acids, in this case leucine or cytosine, are inserted. In some of these cases, These novel mutations, in this case W1282C, actually has reduced function, about half that of the normal state, but it can be augmented further with CFTR modulators. And you can imagine the same sort of approach being conducted, because it has, for the variety of other premature termination codons, determining which amino acids are inserted, which ones might need modulators, and which modulators work best. So we'll now, as we think about tailoring these treatments for individuals with nonsense mutations, we now have a landscape that looks like this. For some people with truncated but partially functional CFTR mutations, we can imagine CFTR modulators being at the cornerstone. Because the mutation occurs late in the CFTR, it's partially functional. And we can use a modulator to augment its function, or perhaps an agent that increases the mRNA levels plus the modulator to get the most out of that CFTR if mRNAs are low. For most people with premature termination codons, they have a truncated and non-functional protein. The CFTR nonsense mutation occurs earlier in the protein, and therefore, even when the protein is produced, it's foreshortened and non-functional. And for these individuals, read-through therapy will be the cornerstone, but can be augmented either with a modulator or in those with low mRNA levels, an agent to increase mRNA. Well, what I hope to have shown you is that correcting nonsense mutations has been a challenging therapeutic target, but we are making progress. Just like for the F508del mutation, where we brought multiple molecules together, with nonsense mutations, we can now achieve this in the laboratory, use them together to achieve levels that are predictive of having clinical efficacy in the laboratory. And while there's much to be done to bring these drugs to fruition, including the preclinical testing, animal models, and ultimately clinical trials in people with CF, we are now very close to achieving that goal, and I hope you'll share our enthusiasm for moving these forward, and thank you for the work that you're doing in this room to make that a possibility. And now I'm happy to introduce Dr. Hastings, who will now take you through splice mutations and novel approaches to do that, and including the progress in other diseases that make us believe that this will become a soon reality for people with CF. Thank you.
Well, thank you, Dr. Rowe, for that really inspiring message. I am thrilled to be here talking with you today about an approach that has shown some incredible results for a number of diseases, and I think has the potential to be a game changer for cystic fibrosis, in particular, those mutations that are not eligible or responsive to the highly effective modulator therapies. And what I am referring to is antisense technology. I started working with antisense technology about 15 years ago, and I was really using it as a research tool in the lab. And then the group that I was working with started working on a disease called spinal muscular atrophy, a severe pediatric neurodegenerative disorder. And they were using a new type of antisense technology to correct gene expression in patient cells and then in animal models of the disease. And I was absolutely blown away by the effects that they were seeing and the therapeutic value that they were getting. That antisense technology ultimately was developed into a medicine for the disease, the first medicine for the disease, and has now been used successfully to treat thousands of children, helping them to sit and even walk, which is truly remarkable for kids that never got to that point in this disease prior. My early involvement in the process completely changed the focus of my work, which is now dedicated to creating more drugs like the one that was made for spinal muscular atrophy. Today, I'll tell you about progress that has been made harnessing that technology for cystic fibrosis. So let's dig in. What is antisense technology? Well, in order to understand that, we first need to talk about how the information encoded in our genes is used to ultimately produce a protein, because that really is where the magic occurs for antisense technology. So the CFTR gene is encoded in our DNA, which is composed of 2 strands of nucleic acids, bound to each other and twisted together and kept nice and protected inside the nucleus. When your cell needs CFTR protein, it turns the gene on, and the gene makes a copy of itself in the form of an RNA. That RNA is a temporary memo that is read or translated into the protein. So how does antisense technology fit into this process? Well, antisense technology refers to molecules called antisense oligonucleotides, or ASOs for short. And what oligonucleotides really means is just a short string of nucleotides, very similar to the ones that make up our DNA or our RNA. An ASO, being short, can be designed to specifically bind to any place in an RNA. You see an example here. It looks kind of like a green comb. And it's base pairing or binding to the RNA based on the sequence. By directing the binding of an antisense molecule in this way to a particular sequence in the cell, we now have a way to manipulate what happens to that RNA. And in this way, we can exercise control and alter the outcome of gene expression. But how does antisense technology, how can it be used to specifically treat a disease and the disease mutations like those in cystic fibrosis? And to answer that, we need to go back to this diagram that Dr. Rowe showed you earlier to think about the core steps. And as you can see, there is an extra step from transcription to translation that needs to occur before you can make your protein, and that is called splicing. You see, some of our genes contain some information that is not needed to make the protein. This information or sequence must be removed before translation can occur. Otherwise, the message will not make any sense to the protein, and the protein won't be made. The pieces of sequence that contain the information for making a protein are called exons, and the additional sequences are called introns. They are removed by the spliced cellular machinery, which were those scissors that you just saw, and then the exons, or the protein coding information, is spliced back together. So the cellular machinery recognizes these splicing sites by a very specific sequence on the RNA. And that makes them vulnerable to mutations. There can be mutations that are created that look much like a new splice site, and that fools the cellular machinery. So here, the cellular machinery is now cutting at the wrong place and splicing together a message that has extra information. And that, as we heard from Dr. Rowe, results in the degradation of that. Our cells have a surveillance mechanism to find RNA that's not correct. And that will cause it to be degraded. So how can antisense technology deal with this? Well, one way that they can do that is because the cellular machinery that splices recognizes a single strand of RNA, we can just make an oligo that binds across that mutation and masks it. And in that way, we can redirect the splicing to the correct sequence and correct splicing. So that's a very simple way that the ASO can correct splicing. So how do we get there apply this to cystic fibrosis mutations? Well, if we consider these 6% of mutations that are not covered by the highly effective modulator treatments, we believe that ASOs can be applied to a number of these, including nonsense mutations and splicing mutations. I'll talk to you first about a splicing mutation that has shown some promise in this area. The C.3849 plus 10 kb mutation is one similar to the example that I just showed you that creates an aberrant splice site that is used preferentially over the correct site and makes a faulty message. We made an ASO to specifically bind to this sequence and block the splicing, correct splicing, and makes a CFTR protein. So how effective was this? Well, remarkably effective. Cells that were isolated from the lungs of a patient with CF have very little CFTR protein activity, as you can see by the white bar there. And they're not responsive to modulator treatment because that RNA is degraded, it's misspliced. However, when we treat with our ASO, we see a dramatic greater than threefold increase in CFTR activity, even without modulator treatment. Similar results have been reported by Batsheva-Karem's group, and in fact, earlier this year, SpliceSense, whose ASO drug is based on foundational work from Dr. Karem, announced clinical trials to begin in late 2022 for CF patients with the 3849 plus 10 kb C to T mutation. More information on the SpliceSense program and clinical development can be found by visiting these posters. There are many additional mutations in CF that have the potential to be targeted in a very similar manner with antisense oligonucleotides through a personalized design. Some of these have already been demonstrated to have ASO options. This has been published by the Amaral group, and more information can also be found at poster 615 from the Karem group. So now let's consider some of the other types of mutations, in particular nonsense and frameshift mutations that might be treated with ASOs. As we just heard from Dr. Rowe, mutations that create stop mutations or nonsense mutations in the CFTR message are rapidly degraded by the cellular surveillance machinery or nonsense-mediated decay. And this, which recognizes the damaged DNA. Any low amount of mRNA that escapes the surveillance is translated into a truncated protein that has no function. So how can ASOs be used in this case? It's a little different case. ASOs can be used to elude this surveillance system and escape degradation by basically binding to the splicing sites on either side of the mutation. And this makes the cell think that this is an intron, it splices it out, and creates an RNA with an intact message, albeit missing some of the information. You can see exon 2 is missing from this, but a partially functional protein would be created. And the idea is that with modulator treatment, you can increase the function of this protein. So my group and the groups of Batsheva-Karem and Adrian Craner all reported earlier this year on exactly this approach for the W1282X mutation. And here, I have the most important slide, does it work? And the answer is yes. Again, looking at CFTR function in cells derived from a patient's lung, there is no activity from CFTR without modulator treatment, without any treatment, and modulator treatment has very low effects. ASO treatment has no effect, but when you add the ASO with the modulator treatment, so you're making this partially functional protein and add in the modulator treatment, you get a dramatic, more than threefold increase, which we think is going to have an impact and prove beneficial in the disease. More details can be found at poster 619 and 638. So at the end of the day, everybody, it's really about hope. As this slide says, and what anti-technology provides is a good deal of hope and potential. There are a number of splice-targeting antisense drugs and medicines in the clinic making differences in diseases like spinal muscular atrophy, Duchenne's muscular dystrophy, and we believe that CF can benefit from this technology in the same way. There's still a lot of work to do, but I couldn't be more encouraged by what we are seeing so far, and I look forward to the advances are going to come in the coming year even, and with some of the trials. So as we build our way to having a treatment for everyone with CF, we can add mRNA correction through antisense oligos to this as a building block towards this goal until it's done. And next we're going to hear from Dr. Rebecca Darrah about another very exciting treatment approach on the horizon, mRNA therapeutics.
everybody. Great. There we go. I'm Rebecca Darrah, and I've got the distinct pleasure of talking with you today about mRNA-based therapies, as Dr. Hastings mentioned. But first, I would like to just take a moment and reflect on something that Casey mentioned earlier, which is the power in this room. And I want to think back on that for a second, because the last time we were all in this room together was 2019, and the FDA had just approved Trikafta as a highly effective modulator therapy for people with an F508del mutation. And I think I wasn't the only one in the room that got a little bit choked up when we saw those data for the first time and just how impactful that was. I want to remind us all that those highly effective modulators, Trikafta and others, are the reason for the 94% in this graph that you've seen several times today. Of course, our focus is on the 6% of people for whom those drugs are not available. And this is a particularly important goal to address, because of those who are not eligible for modulator therapies at this time because of their genotype, a disproportionate number are people of color. And we know that people of color with CF are already much more likely to have faced delays in diagnoses and therefore more detrimental disease courses. So identifying therapies for them remains a really important clinical goal. So I'm very excited to talk with you today about mRNA as a therapeutic approach to treating not just people in that 6% who may not have an effective therapy, but this exciting therapy is really cool because this will be available to anybody with CF, regardless of their mutation. This is a completely mutation agnostic kind of approach. So let's talk about how it works. So first of all, mRNA-based therapies are the reason we are in this room together today. You have all taken one, I think. Judging from the requirements of attending this conference, we have all benefited from mRNA-based therapies. I'm speaking, of course, about the COVID vaccines. So the amount of research that went into developing those vaccines, both before the pandemic and then, of course, in rapid fire during the pandemic, taught us a whole lot about the pros and cons, pitfalls, challenges, successes of using that approach. And those can be applied to cystic fibrosis. So we're going to walk through what this approach might look like and look at some of the preliminary data for how it's going so far. So you've seen this slide before. We've got the DNA with a mutation depicted in red there. And when that's naturally addressed in the cell, it results in a mutation that carries through to the mRNA, the endogenous mRNA that's present. And then that results either in a non-functional protein or oftentimes no protein at all. So the idea of therapeutic mRNA would be that you would bring in a brand spanking new copy of mRNA without any mutation at all, a fully functional copy that would somehow burrow its way into the cell and get translated and make the thing it's supposed to make, the full-length CFTR in this case. And although you would still have the system that doesn't work, this system would be complemented or compensated for by this new mRNA therapy. So there's a couple of things that are challenging about this. That's actually pretty dismissive. There are more than a couple. But one of them is mRNA is very fragile. It doesn't like to be transported around. It doesn't like to be stuffed in cells. It likes to fall apart, honestly. So it's very fragile. So it needs to be packaged into something that can protect it for that journey around your body. One of the most common ways to protect mRNA is to wrap it in this little fat ball called a lipid nanoparticle. And it's really just this protective coating that can cover that mRNA for its travels to where it needs to go. So we'll hear a lot about lipid nanoparticles, and there are different structures to them. You want it to protect the mRNA, but you also want it to be able to unwrap and make that mRNA accessible for translation. There are a couple of different adjustments that can be made to the molecule itself. One is an acrylamide tail, and that helps it be a little bit more protected. The other is an amine head, and the amine head can be placed on the ends of the molecule. And we know that when there's an amine head on the end, it actually facilitates transport specifically to the lungs. And that's an exciting advantage. That's something I like to call a means to an end. Let's see what I'm glad you saw what I did there. So, all right. So let's take a look at how it's working. So one of the companies that's researching this quite heavily is ReCode. And here on the left, we see they've got human bronchial epithelial cells from a person with CF, and they've got them in a nebulizer. So they're taking their CFTR mRNA in that little fat ball, in its lipid nanoparticle, and they're exposing it to these cells just by nebulizer. So the same as if you were to do a breathing treatment or inhaled treatment. So they're not injecting anything into the cells, they're just exposing it via this aerosolized cloud. And then on the right, we see that following that exposure to the outside of the cell, we actually get chloride conductance from those CF cells that looks to be in the range of Orkambi. So in a therapeutic range with this first pass. Now, of course, the goal is to get chloride levels that look more like the range of Trikafta or even higher. But this is really promising to see that even at these earlier stages of development, they're already achieving levels that we would consider a therapeutic dose. So I do know that ReCode is, again, as they all are, fiddling with that structure of the lipid nanoparticle to make it even more accessible, to make it go exactly where it's supposed to go. But this is really, really encouraging, and I hope you're excited by those data. Arcturus is another company that's working on this as well. Here, their lipid nanoparticle is called Lunar. That's their name for it. And in these experiments, they again took human bronchial epithelial cells from people with CF, and this time they put their nanoparticle with the human CFTR in it in the media with the cells rather than aerosolized. And then afterwards, they extracted the CFTR protein, and this is a Western blot on the left. And we see that the cells with either the buffer or a control don't make any CFTR, as we would expect, they're CF cells. But when we put that mRNA in there, they do produce the protein. So that mRNA is getting into the cell and being translated and producing protein. Check that out, not just in a little bit of an amount, but in an amount that's even higher than the non-CF cells. That black band is even thicker than what we see in non-CF cells. So it's getting in there and it's getting translated. But does it work? Well, on the right, we see that when we look at the chloride current from those same cells again, and this is in the presence of a chloride gradient, so this helps to maximize the signal. But we see chloride levels that are equal to or in some cases greater than the non-CF range, and that's what's shown in blue, the non-CF range. So this is really encouraging that those lipid nanoparticles, again, are getting into the cell, they're getting translated, they're making protein we can visualize, and that protein is functioning. Now, if you're following along, and I hope you still are, I hope you're asking next, what about the mucus, Becky? What's going on with the mucus? So we know that one of the big challenges for CF is that lipid nanoparticle with its cargo in it doesn't just have to get to the airway cells, it has to cross a really big barrier, and that's the thick mucus we all know is present in people with CF. So for that, we can turn to the CF ferret. And for those that don't know, the CF ferret actually does produce mucus that's similar to the mucus that people produce, people with CF, especially when it's not being given a modulator therapy. So in this picture here, we see that this is the airway of the ferret, and on the right, you can see they've actually stained for the presence of that mucus in the bronchus, so we can see evidence that that mucus is present. So this is a pretty slick experiment. So what they did here was rather than use their lipid nanoparticle with CFTR in it, that lipid nanoparticle was delivering something called Cre recombinase, or simply Cre. And what that Cre does in this specialized ferret, if it gets where it's supposed to go through the mucus into the cell, it will turn a red cell into a green cell when exposed to this special camera in this imaging system. So that's pretty fancy, right? This is kind of complicated genetic stuff. So this Cre recombinase gets into the cell, and it will turn the red cells into the green cells. And you can see in the trachea and the bronchus there the number of green cells. So this was delivered via aerosol to this ferret. So this ferret takes this in via aerosol. The mRNA gets where it needs to go through the mucus into the cell and is able to function. And this way we know it functions because of the color change. So this is really exciting in vivo work that I hope you're excited to hear about. And I'm thrilled to say that these 3 companies are all planning clinical trials for 2023 for mRNA-based approaches. Woo! Yes. And that's just an amazing thing when you think about the where we've come on mRNA-based process, in large part, of course, because of the pandemic. But to be able to apply that here is really remarkable. So we look forward to hearing about some of these results, perhaps even at next year's plenary, on how they're going. So I want to take a step back and reflect. You know, I've been involved in CF basic science research for almost 30 years. And I've been a genetic counselor for a little more than 20 years. And when we first started thinking about it, the first conversations I had about genetics and CF were almost entirely focused on inheritance. And, you know, we talked a lot about just the basic concept that as an autosomal recessive disease, 2 people who are carriers for CF would have a 1 in 4 or a 25% chance of having a child with CF. And, you know, there was all right, we've got that. That seems to make sense. And there were a lot of different ways we could explain that. And then the really advanced question, you know, the question that really stumped everybody was thinking about the fact that the healthy siblings of someone with CF would have a 2 out of 3 chance to be a carrier. And that was the question that we convinced all of the residents was going to be on their board exam. And, you know, we made sure they understood when it's 2/3 and when it's 1/2. But that's really kind of where the genetics conversation ended for a while. I hope you can appreciate today the types of genetic concepts that we've talked about are really complex. There's quite a bit going on, and even for people who are geneticists, such as myself, there's a lot there, and it can be quite confusing. These are some quotes from some of the patients I've interacted with in my clinic, and other genetic counselors have as well. You know, I just want to take a moment. I bring these up because I think they highlight just how confusing some of these concepts are. There's still confusion about gene editing, which we haven't talked about. You take a moment and read them, sure. People are still picturing a surgery for that, I think. The nomenclature, this is one that gets me. You know, there are at least 6 different ways lab reports report back F508del. And that's really confusing to families with CF. You know, it looks completely different. Those 2 genotypes don't look the same at all. And that's something that can be difficult to explain. I'm not even going to get into the T-tract genetics. You know, this is complicated for all of us. But you just need to know that the 5T variant refers to a string actually of T's in the DNA sequence. And it wasn't until somebody asked me if that's related to a low T cell count that I realized those things sound similar. So I think, you know, being cognizant of the ways these things can get confusing is really important. Finally, some of the words that we use, such as nonsense, have multiple meanings. You know, it can sound really dismissive when your doctor says your mutation is nonsense when you don't have the right context for that. So if you are lucky enough to have a genetic counselor in your clinic, hopefully you have come up with some strategies or you've thought a little bit about how to explain some of these complicated ideas. If you do not have a genetic counselor in your clinic, first of all, I hope you consider it. You know, I know I'm a little biased, but I hope that you can consider it. But I will also offer you an analogy that might work for both your own understanding and perhaps something that you can bring back to the clinics with you and help in explaining this to the families. So we're going to walk through a gene as really a recipe. And since we're here in Philadelphia, we're going to make a cheesesteak. Has anybody tried a cheesesteak in Philadelphia yet this time? Okay, good. All right, so you'll be with me with this. So if you've looked up a recipe anytime in the last 5 years, maybe online, you'll notice that you don't get just a recipe anymore. You get a whole long list, right, of the market you're supposed to shop at, and the smell of your grandmother's kitchen when she made the recipe, and all of this sort of filler that's in there. And if you read through the recipe, you see there are ingredients hidden in there, but it can take a little while to dig them out. So we can think of this as similar to the DNA, the RNA, and the protein, right? So the DNA becomes the entire recipe. The RNA becomes the ingredients list that can be excerpted from that recipe. And the protein then is the thing that you make, in this case a cheesesteak or the CFTR protein. And we can use this to help understand how these mutations might affect something. So for example, a nonsense mutation, which we've heard a lot about today, is as if there's a period in the middle of the ingredients list, and then no one shops beyond that. There's nothing to modify. There's nothing to address. There is no cheesesteak. That's disappointing. There's nothing there. Whereas a splice site mutation might be where there's extra things in the ingredients list. These are actual words that are in this recipe, right? But they don't belong in my ingredients list. I don't want to ask the butcher to slice you. You know, that's not something that's going to be helpful. Sometimes with a splice mutation, there might be missing things from the ingredients list. And in this case, I can actually make a grilled cheese sandwich. It's not the cheese steak I set out to make, but it might still function if I'm hungry, right? This might still do something. So part of the point is that some variants are more detrimental than others. Some produce something different that might still function, but some produce nothing at all. And we can think of that same concept with deletions. So for example, if I delete the word green from my recipe list, I still have half a bell pepper sliced, and it doesn't really matter what color it is. I'm going to get a pretty good cheesesteak that I think will do its job. But if I delete the word pepper, yeah, I've got half a green bell. That doesn't make any sense. That's not something I can use in my cheesesteak recipe. So I think hopefully this makes sense to you, and you can perhaps use this with families on how to convey these concepts. Finally, we can also think of the therapies in this sort of sense as well. So again, we haven't discussed gene delivery or gene editing in this plenary, but we can think of that as either bringing an entirely new recipe to the table or doing a spell check on the one that we have and editing out any mistakes that are there. We have talked a lot about RNA-based approaches. The nonsense approaches might be akin to reading through that period or whiting out the period and keep reading on the ingredients list. Maybe the ASO approaches that Dr. Hastings talked about are similar to putting a big block over the area of the recipe that you don't want to be transferred over to the ingredients list or adding something back in. And then finally, the modulators are some way to make that cheesesteak taste a little bit better or that protein work a little bit better. There's just been an immense amount of progress in the field of genetic-based approaches to therapies over the last year or so. And I'm really excited to see that CF research is right in the foreground of that. We're going to see many more of these headlines with the word cystic fibrosis in them. It's a very, very exciting time to be part of this and to see those successes. We've talked about, you're already familiar with modulator therapies, but today we've talked about new approaches for nonsense mutations and also for splice mutations. I've talked about mRNA replacement, which again would be an approach for everyone with CF. This is not mutation dependent. And the idea is that between all of those, with all of those put together, we will achieve 100% CFTR restoration for all. And thank you, yes. And we can assure families like the McClains that we in fact will not give up until it's done. Thank you.